interventional pain treatment. I have no financial declarations to make, but one disclosure that I come from the basic sciences. I'm a neuroscientist. I'm not a physician and not responsible for patients, but do try to work on or understand mechanisms of pain, particularly neuropathic pain, and that's what I'm going to be discussing today. And in particular, what I'd like to try to get across is an idea for a kind of interventional treatment targeting the dorsal ganglion that is not currently being used, and I think probably deserves a try. And I think you are the people that if this is ever going to get a try, you should be the ones who would be doing it. I think that this approach that I'm going to try to explain, convince you is worth a try, is likely to be effective for a number of major neuropathic pain states. And as I mentioned, my perspective is that these are similar in many respects, including herpes zoster and postherpetic neuralgia, CRPS, trigeminal neuralgia, which we just heard about, phantom limb pain, and radicular low back pain, which is actually the thing I'm going to focus on today. The reason that I'd like to focus on radicular low back pain is that it illustrates a central challenge in pain science and medicine, a challenge that has been building and building and building, and I think we're at something of a crisis now. I take these statistics on the leading causes of disability in medicine, in all of medicine. And you can see that number one on the list 30 years ago was low back pain. And the most recent publication of this series, just a couple of years ago, continues to have low back pain as the number one cause of disability in all of medicine. It's interesting to see that the number two is also a pain condition. So somehow or other, we haven't been doing very well in dealing with these severe, very common causes of diseases, causes of disability. And I add to that, of course, the disaster of the misuse of opiates, particularly in North America. It's to the extent, at least this is the way I see it, maybe I'm too pessimistic, that we're even entering a period of despair. We start seeing senior voices in the pain field saying that maybe we should give up the whole idea of pain relief and instead be teaching patients how to live with their pain. I, as a scientist, am not willing to accept that approach. I think we just have to continue working and applying the great increase in knowledge that we have from the basic sciences on mechanism and translate it into the world that you people live in, where you're actually dealing with patients and trying to help patients. But just as one example, here's a recent paper from two leading pain clinicians, both very smart, who say, must we reduce pain intensity to treat chronic pain? Do you see what they're saying, actually? Let's give up on relieving the pain. That's the end point we really want. And instead, making people accept their pain or live with it a little bit better. I'm going to make two main points during this presentation, putting across this idea that I'd like you to consider. The first is that radicular low back pain, and the other conditions that I mentioned as well, I believe at least. The radicular low back pain is driven primarily by afferent discharge, afferent activity, sensory activity, electrical impulses that are generated ectopically in the dorsal ganglion. Now, when I say ectopic, the meaning is that sensory input is normally coming from the skin or peripheral tissues, and if it's coming from any other location, like the dorsal ganglion, that's ectopic, it's the wrong place. And secondly, the dilute membrane stabilizing drugs. I'll define what I mean by that shortly. Dilute membrane stabilizing drugs applied intraforaminally onto the dorsal ganglion in the intervertebral foramen should suppress the discharge and the pain without blocking the conduction in intact sensory fibers or intact motor fibers. So those are the two messages I'd like to get across and try to demonstrate to you. But first, let's begin with the system itself. So I have here a drawing on the left, on the bottom at least, of the peripheral nervous system entering the spinal cord and then the spinal cord up to the brain. So stimuli are normally applied to peripheral tissues where there are sensory endings which transduce a force or a temperature or a chemical stimulus into electrical impulses which run up the nerve past the dorsal ganglion into the spinal cord, from there up into the brain, activating a pattern of activity in the cerebral cortex that is called the pain matrix, or some people prefer the fingerprint or signature of the activity. And this pattern is supposed to actually be the physical, the neural basis of the sensation of pain. Just for fun, if you want to read something piquant and interesting, a paper we published recently that argues that this pain matrix may have nothing to do with pain at all, and that pain may be something that's perceived in the brainstem, you might want to look at this. Well, can't really see this reference right here. Be that as it may, the dorsal ganglion is the location in which the cell bodies of the peripheral nerve fibers reside. So the DNA, RNA, the generation of all the molecules that support the peripheral nerve are in the cell bodies in the ganglion, which is paraspinal. So that's the dorsal ganglion, and of course it appears on both sides of the foramen between adjacent vertebrae. That's what the ganglion looks like. Those little grapes are the cells, and the little wormy things are blood vessels. And you can see the nerve up on to the right, labeled N, and the dorsal root on the other side. A variety of different kinds of cells with different properties, some sensitive to heat, some sensitive to mechanical touch, light or strong, and a variety of different sorts of signals that together, in sort of like a symphony, produce the natural sensations that we all feel when there's our natural stimuli. On the upper right is a drawing, schematic drawing of a primary sensory neuron. You can see it's got a cell body, a small branch that breaks into two, for making the peripheral nerve and the dorsal root. This comes from the famous Kendall textbook of neuroscience. It's very misleading because the cell bodies are quite small, 40, 50, maybe a little bit larger microns across, where the axon can be very long. So if we're talking about the axon from the dorsal ganglion here to the end of my finger, that axon is 1 meter long, and to my leg it's even longer. If we actually draw this in scale, you see the cell body? That's the size of the cell body. That's the 50 microns. And the dorsal root axon is the thing that's going back and forth all the way up into the spinal cord, and the axon, the peripheral axon, is the one that's going all the way down to that sensory ending at the bottom. So as far as the cytoplasm is concerned, a tiny, tiny fraction of the cytoplasm of sensory neurons resides in the cell body. Almost all of it resides in the axon. And if you have a peripheral nerve injury, such as that, a nerve injury almost anywhere along the nerve, you're actually, in terms of the cytoplasmic volume, the actual volume of the cell, you're cutting it in half. And of course you would expect that cells would respond, would change as you slice them in half. And that's what happens when there's a peripheral nerve injury of one sort or another. The cell body in the ganglion does, and its axon, does respond in several ways. The most important one, I suppose, is that both of these sites, the neuroma, the area of the nerve injury itself, and the sensory ganglion, begin to fire impulses spontaneously, without any stimulus at all. And in addition, the ganglion and the sensory ending, the neuroma, become abnormally sensitive to mechanical tapping, that's the Pinel sign, and also to various chemical substances. So we have ectopic firing coming from compression sites, demyelinization, frank cuts, and entrapment tumors, and whatever. So lots of different things can injure the axon, and the neuron will respond in much the same way, becoming hyperexcitable in terms of generating impulses at both the injury site and in the ganglion. And here's just an example of what this looks like when we record from animal nerves, and it's very similar when recordings are made from humans. You can see, for example, some fibers will go brrr, just fire spontaneously forever, for as long as they last. Sometimes in bursts, you can see this one here. Sometimes an irregular pitter-patter. And if you look at the nerve injury site itself, you can always find very tiny little spots, or actually many little spots, where individual axons have been cut and become mechanosensitive, thermosensitive, and chemosensitive. Now, what you might not know is that very much the same things occur in the dorsal ganglion. It also becomes abnormally sensitive to various stimuli and a source of spontaneous activity. And once again, the impulses run up into the central nervous system and give us a sensation of pain, neuropathic pain in this case. This actually has been known for a very long time. The first publication I know about it, of the ganglion being a source of abnormal impulses, comes from 1974, and Pavlov and I repeated this and proved that it really is so just a couple of years later. But that's a long time ago. It's 40 years ago that this has been known. But it hasn't really been a target for interventional treatment until very recently. I'm not going to go into the details of this experiment, but the bottom line is to compare which is the major contributor of ectopic discharge and mechanosensitivity. Is it the neuroma that we always think about, or the ganglion? And in fact, the results of the experiment show that something like 3/4 of the activity, and the ectopic activity, the abnormal firing that enters the dorsal ganglion into the central nervous system, is coming from the ganglion rather than from the nerve injury. We prove that by making cuts along the way and seeing how much of the activity vanishes. Now, we have a pretty good idea of why this happens. That is, why the neuron becomes electrically hyperexcitable. And it has to do with something that I'm sure you all learned and perhaps even forgot about basic science and impulses and sodium channels and potassium channels. But these things make neurons fire electrical impulses. And we're talking about a change, faulty regulation of the excitability of the neuron. And there are three basic kinds of faulty change in the regulation of excitability. One is a change in gene expression, another in protein trafficking, that is, where the proteins responsible for the impulses go. They're all made in the cell body, but they have to go somewhere else in order to do their job. And channel kinetics. So let me show you what I really mean by that. So the sodium potassium channels responsible for making impulses are generated, DNA, RNA, protein, generated in the cell body, and they're transported along the axon down to its end, down to the sensory ending. That's what normally happens. And the regulation of how much of this protein should be sent down and which kinds depends usually on growth factors in the periphery that signal back to the cell body. So the cell body is always being controlled so that it doesn't make too many or too few channels, so that the excitability, responsiveness of the neuron remains within normal bounds. However, what happens if there's an axonopathy, if there's an injury to the axon, or some other change that alters the regulation? Well, let's look at the situation of a nerve being cut. We're now disconnecting the feedback, the control from the periphery, the growth factors from the periphery, from the channels that are being sent down. What actually happens is that the presence of sodium channels accumulates at the cut nerve end. You can see this little balloon here, where the arrow is. So these channels simply accumulate at the cut end. They also accumulate with the cell body. You can imagine water flowing in a hose, and you squeeze the hose, so the water just accumulates, the pressure accumulates proximally. This is what it looks like if we use an antibody that can visualize for it. We can see the antibody or a fluorescent dye that's attached to the antibody. So you can see on the top that's an axon that has been cut. You can see that yellow arrow on top, that's where the cut was. And when we stain this section for sodium channels, that is the molecules which are responsible for making electrical impulses, you can see a very substantial accumulation of these channels at the cut nerve end, just for this simple, easy to understand reason that I mentioned. This is what it looks like within a nerve. Now, we're all familiar with this very famous picture or idea that if we've had a disc herniation and damage to roots or even spinal cord, usually roots and ganglia, that even expert radiologists are not able to sort these images into the pile of people that have pain and people who don't have pain. And this has always been a terrible mystery, but I don't think it's a mystery at all. I think what it really means is it's not the injury that's causing the pain. What's causing the pain? It's the accumulation of sodium channels. So if this individual here, who has no pain, if we could image, if on top of this CT or MRI, if we could add an image of the sodium channels that were present, we would probably see nothing there and lots here. And I think that is going to be the marker of whether this individual has pain or not. But we just don't have the technology yet to do that. Now, so far I've been showing you experiments on rats. Here's a recent publication where dorsal ganglia were excised because in cancer patients that had to have the low back pain surgery to remove the tumor. So at the same time, ganglia were excised and recorded from, in the same way that I showed you from the animals. And in fact, this is human ganglia. Some of the patients had pain, some of the patients didn't have pain. And you can say the patients that had pain had lots more abnormal activity in their dorsal ganglia than the ones that had no pain. And to look at a similar thing, there are some human genetic conditions, rare genetic conditions, which cause excessive pain. I'm referring at this point to work from Steve Waxman's lab on one of the sodium channel types. It's in the news a lot, NAV1.7. So a particular mutation in this sodium channel causes very severe continuous pain. And we can make a rat's dorsal ganglia, or in vitro, we can make dorsal ganglion cells express this abnormal sodium channel. And when we do, when Waxman does, you can see that the cells are much more excitable. The same stimulus, instead of giving 2 impulses, is now giving, what is that, 7 or 8? Or here, it's now giving perhaps 15 or 20 impulses, where it should only be giving a couple. So these same conditions, same changes, and we're talking about point mutations in a large protein, the same condition, mutation that causes this very severe pain condition in humans is causing more impulses in the dorsal ganglia. Again, I'm trying to connect between the symptoms that you see and the physiology that we know is happening in the nervous system. Okay, so what's all this got to do with axial low back pain and leg pain, the shooting or extension of the pain into the low back when present? Well, up there is a picture that you all are familiar with. Spine, muscle tissue on the lower back, and the ganglia, and peripheral nerve is here, of course. Whoa. Forward too fast. Whoa. Okay, there we are. The peripheral nerve and dorsal roots. And there, we usually talk about 3 different kinds of pain. Normal pain, when normal nociceptive endings are activated. That, of course, can be a cause of pain in the low back. We're also expecting that if there's inflammation in this low back tissue, that will sensitize sensory endings and increase the sensation of pain in the lower back. And in fact, one of the locations where this inflammation could be coming from is nucleus pulposus material that can be leaked into the tissue of the low back and cause sensation pain in the low back. So where would that pain be felt? It would be felt actually in the low back. And that's due to normal or inflammatory conditions. And to a certain extent, it could actually be caused by a neuropathic condition. So if, for example, there's a herniation of the disc, this is affecting mostly the dorsal ramus, that is the nerve fibers that serve and innervate the low back, you would expect that also to be causing pain felt in the low back. But how about radicular pain? What might cause that? Well, radicular pain presumably would be caused by activation of the cells which send their axons down the leg. So the impulses would be coming from fibers that actually innervate or serve the low back. So the dorsal rami will give you sensation in the low back, and the ventral rami will give you sensation in the leg. And all of them can be neuropathic, inflammatory, or normal, depending where the inflammation is and where the neuropathy is. So this analysis would say that the low back pain is coming from tissues, from innervation of the low back, and that the leg pain is coming from ventral ramus. Now, there is another possibility that has been in the literature for a long time, which argues that the normal and inflammatory pain should be coming from dorsal ramus fibers, from the low back. And the leg pain may simply be referred. And there is some basis for that. For example, if one injects hypotonic saline into tissues of the low back, one can get reports of pain that runs down the leg. So perhaps the radicular pain is simply referred pain, and the treatment would be different. It would not be neuropathic because there's no involvement of the ventral ramus. So is radicular low back pain, the shooting pain down the leg, is it neuropathic or is it referred nociceptive? And it matters because the treatments would be different. If it's an inflammatory pain, it should be treated with NSAIDs or with steroids or anti-inflammatory drugs. And if it's neuropathic, it should be treated with the various drugs which suppress abnormal activity, sodium channel blockers, anticonvulsants, and whatever. So how can one know? So let's consider this situation. Straight leg lifting, pain, Lasek sign. Nordin et al., quite some years ago, recorded from the peripheral nerve with a microelectrode. Now, let's say that this is referred pain, that the impulses are coming from sensory endings, either normal or inflamed, in the lower back. And the impulses now are running from there into the spinal cord and up to the brain and giving a sensation of pain in the back. The alternative that we've just been talking about, that the ganglion is the source of the impulses, would suggest as follows. So if we have a normal sensory ending, the impulses can only go in one direction. But if the impulses are generated in the ganglion in the middle, they can go out, and they can also go into the central nervous system. They'll go from the middle in both directions. Now, it's possible to test that, to ask that question. So if the impulses are going in both directions, we should have impulses that are generated in the ganglion that run out to the leg, out to the foot, along one of the nerves in the leg, which could be picked up by the electrode. So we have an experimental question that we can test in humans with radicular low back pain evoked by straight leg lifting. So what is the result of the experiment? Or of the observation, the clinical observation? And here we have it. As the pain comes on, lots of electrical impulses are recorded in that distal electrode, almost a meter away from the ganglion. So the impulses must be coming from the middle of the nerve, from the ganglion, and going in both directions. So we have here really a very good piece of evidence that radicular low back pain is at least a major contribution. The feeling of the leg pain, not the concurrent back pain, the feeling of the leg pain is originating in the dorsal ganglion. And that might be a way of treating it. Remember, this is the #1 cause of disability for the last 40 years. I recommend this paper that goes into a proof of that with a tricky surgical procedure. But together with Silvio Brill and Dufresne, we published quite recently a bedside test where one could actually check with a very simple sensory testing whether it's likely that the pain is coming from the ganglion or from the low back tissue itself. I recommend that you read that paper in pain. So what I hope I've convinced you of is the dorsal ganglion ectopia, the ectopic firing generated in the dorsal ganglion, is a key driver of low back pain, radicular low back pain. Now, the second thing I'd like to convince you of is there's something to be done about it. That is the dilute lidocaine or other membrane stabilizing drugs. When I say membrane stabilizing drugs, I'm referring to drugs that suppress the impulse initiation. For the most part, they're sodium channel blockers, but not only. Most of the ones I'm going to be describing are, in fact, sodium channel blockers, like lidocaine local anesthetics. And the point is going to be, or what I'm trying to demonstrate now, and we're getting near the end at least, that suppressing these drugs at low concentrations, and I stress low concentrations, selectively block the initiation of impulses, the generation of impulses, at concentrations that are far too low to block the propagation of impulses. So if the impulse is already generated, you have to use high concentrations to block them. That's what the dentist does when you inject 2% lidocaine into the nerve, and we talked about nerve blocks yesterday. But low concentrations are enough to stop the generation of ectopic firing in the ganglion, in the neuroma as well, particularly in the ganglion. So let's look at this experiment where we've, in a rat, injured the peripheral nerve. We're recording now from the point R, which is axon central to the neuroma, central to the nerve injury site. And what you can see is there are lots of activity recorded from those nerve fibers, a lot of it bursting. Those are the pictures you see. And each of these two small arrows below is the injection of very low concentration, a very small amount of lidocaine into the peripheral, into the circulatory system, an IV injection. And you can see with the first one, you can see that the firing rate has gone down, the bursting is less, and with the second one, the bursting has stopped altogether and the firing is gone. So we've silenced the abnormal spontaneous firing. However, the axons are still able to conduct impulses. How do I know that? Because we stimulate electrically here before the block, and after the block, the nerve conduction is identical. So this actually just shows us the dose-response curve for blocking the abnormal firing. So once we get up to about 3 milligrams per kilogram IV, we've stopped all of the ectopic firing. The dorsal ganglion is a little bit more sensitive than the neuroma. By the way, in emergency rooms, if someone comes in with a horrendous neuropathic pain, they often give IV lidocaine. And I sometimes ask people, physicians, why does the IV lidocaine work? And I sometimes get the surprising answer, well, everybody knows lidocaine stops impulse conduction along nerves. That's what the dentist uses. But does that make any sense? If you gave enough lidocaine to block nerve conduction in the body, you would immediately stop the heart and stop the respiration and kill the individual. We're talking about concentrations that are something like 10,000 times lower than the 2% lidocaine that's required to block. Now, that's the curve for lidocaine, a local anesthetic. But it's not just lidocaine. So on the left, I'm not sure if you can read it, that's carbamazepine, which is one of the anticonvulsants. In fact, most of the anticonvulsants will do the same thing. Naxolotine, which is an antiarrhythmic, does the same thing. Amitriptyline, which is sold as a catecholamine reuptake inhibitor and an antidepressant. If you look in the pharmacology books, you'll also see it's a sodium channel blocker. And it stops ectopic firing at concentrations far, far lower than what would be required to stop nerve conduction. Gabapentin, very much the same thing, even though it's sold again with a different mechanism in mind. So virtually all of the systemically available adjunctive drugs that have some efficacy for relieving neuropathic pain are in this category of membrane stabilizers, no matter what the sales department uses to promote them. They all stop ectopic firing at very low concentrations without blocking nerve conduction. So if we've got such great drugs, what's the problem? Well, the problem is the adverse side effects. The systemic delivery of any of these drugs gives basically the same side effects: sleepiness, nausea, vertigo, lethargy, foggy thinking, central nervous system problems. What's the solution? Well, in principle, one should be able to make any of these drugs, gabapentin or lidocaine or something, in a form that doesn't cross the blood-brain barrier and doesn't get into the brain. It only works in the periphery of the ganglion, the nerves where we think the problematic impulses are coming from. The cardiologists had the same problem exactly in the 1950s and 60s with beta blockers. They just peripheralized the beta blockers so they couldn't get into the brain. And half of the people in this audience are probably taking these drugs every day to great effect with very minimal side effects. Another idea is to get blockers that are specific to the sodium channels that are specific to the dorsal root ganglion. There are many kinds of sodium channels. Some of them only appear in the ganglion. There's a lot of money being invested in that in these past years without much success so far. Or work on potassium channel openers. The problem is that all of these things require big pharma and huge amounts of money. It's not something that you folks can do. But there is a simple, relatively simple interventional approach that could in principle be used. It looks like this. Sensation from peripheral receptors is from the skin and other tissues, and cell bodies in the ganglion. The impulses run past the ganglion into the central nervous system. That we already said. And the motor fibers go out. But we're now saying that the dorsal root ganglion is an important initiator, generator of impulses responsible for radicular low back pain and the other long list of important neuropathic pain conditions. So those impulses are coming from the ganglion. So if one could apply the very dilute concentrations of local anesthetics or anticonvulsants or antidepressants to the ganglion, that ought to stop the ectopic firing without blocking either the normal sensation or the motor control. We actually tried that in rats. In this experiment, we're actually recording impulses generated in the ganglion and also impulses that pass the ganglion through conduction. Without going into too much detail, and that's the location where we've simply injected through a catheter a very small amount of dilute local anesthetic. And what you can see is that there's a very substantial decrease in the ectopic firing, but no decrease in the through conduction. So we haven't blocked nerve conduction, but we have blocked the generation of ectopic firing. And if we look at various concentrations used, there's a therapeutic window here in which we can quite reliably stop all or most of the ectopic firing without blocking nerve conduction. And if one works within this therapeutic window, one ought to be able to relieve neuropathic pains that are generated in the ganglion. We've tried this behaviorally with animals as well. I hope I'm not running on too long. Okay? Okay. So here is one of the common models of neuropathic pain in rats. So the nerve, peripheral lymphatic nerve in this case, is cut across. We're testing the sensitivity of the rat when we touch his paw. So normally it takes about 25 grams of force before he'll withdraw his paw. But after the nerve injury, it goes down to about 2 grams. So this is tactile allodynia. So we're generating tactile allodynia. And now we insert a pump into the animal that has a catheter that goes up to the ganglion. And you can see that quite soon after, a day or so after inserting this pump, that the allodynia goes away. The animal's responsiveness normalizes. And now we remove the pump, and within a day, the allodynia is back. So we're doing in rats what I'm hoping someone might want to try in people. We do have a bit of evidence, and this is from work of a friend and colleague of mine, Pascal Basso, who tried this for phantom limb pain. So the idea was simply to inject, first we tried full strength lidocaine, and then we tried with dilute lidocaine around the ganglion associated with the phantom limb pain. And the bottom line was nearly all the amputees reported greater than 75% relief, transient of course, of their phantom limb pain for hours or perhaps a day. I simply don't buy that an injection of lidocaine can give you pain release for a month or two. Something else has to explain that phenomenon. In any event, this already shows, I think, that phantom limb pain is generated as one more neuropathic pain. Here the impulses are coming from the ganglion, and has nothing to do with cortical remapping, which is a theory that's become very popular. In any event, repeated injections on the ganglion kept us going for about 2 weeks, but we didn't have the facilities for pumps or anything longer than that. So a word about neuromodulation, not using blockers, but using electrical stimulation. So we all know about TINs and stimulation of A beta fibers, which would auto-suppress painful activity. That was the original TINs that was developed a very long time ago. And very soon after that, became dorsal column stimulation, which is now called spinal cord stimulation. And less TINs, the more you stimulate them. We've never gone to a day or continuous electrical stimulation. But my guess is that it could stop the firing for a while. In any event, this method does have some efficacy, the same way that TINs has. But my proposal, my guess is that rather than adding impulses in the hopes that this will cover the pain, that if one were to deliver dilute membrane stabilizing drugs to the ganglion, one might get a much better and more effective effect. So what is actually being proposed? Place a catheter in the ganglion, test if it actually works for whatever the condition is, using a port, and if it does work, then put in a pump. And since very low concentrations are going to be enough, you could actually probably fill this pump with concentrated lidocaine and use such a low pumping rate that one refill might work for a month. Who knows? Now, if it works, one could then, or one would hope that the companies would invest the same efforts that have been invested in modernizing electrical stimulators. The pumps we have are very primitive. The problems that, I know that doctors don't like using these pumps, but the main problems are that the tubes get pulled out, they're not anchored properly, or they're not elastic enough, and that refilling them is a problem, although EpiPen, there should be refilling procedures that would be safe and easy to use. I'm sure that a lot of improvement could be made in pumps if this proved to be effective for particular low back pain, for example, to say nothing of all the others. And so maybe it's time for someone here to start a startup and bring pumps into the modern age. There are other interesting approaches for delivering low concentrations of lidocaine and other such drugs. One, and with this I really do end, is something that's more futuristic, but a biopump. The idea would be to take a little piece of autologous tissue from the patient himself, a little piece of skin, for example, to infect it, this piece of skin, with a virus which delivers the DNA for manufacturing a lidocaine-like peptide. That is a peptide that will be a channel blocker that will reduce abnormal firing. And then with these little shims of tissue, you would just load them into a syringe and inject that into the foramen. And that, in principle, ought to do the job, either if the impulses are mostly coming from the ganglion or if they're coming from the periphery. Skin doesn't hurt, muscles don't hurt, joints don't hurt, bones don't hurt. If you want to know about pain, you have to think nerves. Nerves hurt. And the primary question you should be asking, what is the source of the impulses that are causing pain in this particular patient? And try to attack the place at which the impulses are generated, rather than blocking the nerve, which requires high concentrations and problems. Neuropathic pain is a disease, and as I mentioned, I think it's not exactly unitary. There are differences between the conditions, but they have the same principal abnormality, which is faulty regulation of neuronal excitability. This is a nerve. This is a bowl of spaghetti, and please remember that they're not the same. Thank you very much for your attention.
I call it brainstorm. We have ideas coming in our mind. Any question? Yes, please. I was expecting.
Yes, thank you for this really beautiful lecture and sharing all your ideas with us. We met before and we discussed about the DRG and the application of lidocaine. We did a lot of work in DRG stimulation, especially if you look at the low back. You see that there are DRGs who are more attractive and DRGs who are less attractive. For example, stimulating on the DRG of L2 or T12 gives more effect on the low back as stimulating one of the others. In your experiment, you have completely control, of course. If you do it in a rat, you know where you make the lesion, you know which DRG you have to attack. How do you see this in humans, where there's a lot of crosstalk between these DRGs?
You know, I think the problem is that we don't really understand why adding electrical impulses, and it's adding electrical impulses not to the pain-provoking fibers, but to the A beta fibers. If you turn the current up high enough to stimulate the nociceptors, that would really hurt. We don't really know why this suppresses pain. If it's the gate control theory, which I think is still the best hypothesis, although there are several other hypotheses, then you have to get the signal into the spinal cord. And normally you would do it sort of in adjacent or overlapping dermatomes, overlapping sections. So it may be that you don't want to stimulate in the ganglia of the pain, where the pain is felt, but adjacent to that, where you would get a better suppression effect or gate control effect in the spinal cord. So I'm only guessing that might be the reason. But since we're missing a key part of the puzzle, why does the stimulation reduce pain? I think it's not a reasonable hypothesis. But with suppressing the actual generation of impulses, I presume it has to be in the segments that are appropriate to the leg. 4, 3, 5. But if it worked, we would know that immediately. If this ganglion didn't work, you'd just try the next one along. But I still think the principle of suppressing the abnormal activity, rather than adding to it and hoping that some interaction will reduce the pain, I think it's worth trying. Now, I realize that the electrical stimulation is being sold, it's been developed, the engineers are doing fabulous things with it. And I guess today that's the thing to do. But I think that this method might be more effective, and if someone would give it a serious try, then we would know whether it's worthwhile investing in this direction. I hope that's an answer.
Please.
Sure. Thank you, Doctor, for your lecture. It's amazing. Especially, it concentrates on the physiology of pain. You mentioned that to decrease firing or stop firing from the dorsal ganglia, it can be by injecting a low dose of local anesthetic around, or by, my colleague said that, dorsal root stimulation. So, all of us know that neuroplasticity is central. Central neuroplasticity that leads to chronic pain and sensitization. All of us in the last decade are concentrating on pain gate theory, like dorsal root ganglia stimulation. It goes with the pain gate theory to suppress central stimulation or neuroplasticity. The new thing is to inject local anesthetic around. We are not stimulating. We are not activating the pain gate. We are suppressing the firing to the dorsal root. So it's confusing.
I think, and I'll apologize for saying these things, that your opening statement, that we know that neuropathic pain is a central phenomenon, that it may have started in the periphery, but it's become centralized, and now the problem is in the central nervous system. I think that this is a falsehood that has taken hold in the medical community when patients come in and have a terrible pain that I can't treat. So I say, well, I blame it on the fact that it's become centralized. Had you come to me earlier, I may have been able to help you, but now I can't. I think there's very little evidence that impulse generation within the central nervous system is causing any of these neuropathic pains. I mentioned the one example of phantom limb pain where people believe it's a cortical reorganization. I think that never made any sense. The data simply didn't say that. And in fact, if you block the ganglion, you'll get rid of the phantom pain. You don't even have to block the ganglion. It's enough to do an epidural block, as in childbirth. And that would block the signal from the ganglia up to the brain. And just ask the question, does epidural block stop phantom limb pain? You don't expect it to block phantom limb pain for a month or for a year. It should block phantom limb pain for a couple of hours. But if it does, that's proof that it's got nothing to do with, that the pain is not generated in the cortex, it's generated in the periphery. The same goes for fibromyalgia, for what it's worth. We have lots of evidence that there's a peripheral neuropathy, both from recording and from histology, and yet everyone believes that fibromyalgia is something that's generated in the central nervous system. That could also be known if we just do a peripheral block, an epidural block, a fecal block, and see whether for the next half hour or hour the fibromyalgia pain goes away. I bet it does. So one can get to believe almost anything. I don't think there's anything that human beings cannot accept and believe, because that doesn't make it true necessarily. You need evidence. And it also has to be logical. There are surprises sometimes. Things generally are logical. Thanks a lot, Matt. Thank you.
Great. Thank you.
I apologize for taking too much time.
He's available for anyone questions outside during lunch break. Yes, please.
Thank you, Marshall, for giving me 5 minutes left for my lecture.